Thank you. I need to thank Francoise and Sharon for the invitation. They wrote me and asked me to give an overview on cell and gene therapies and immunotherapies toward HIV cure, and I was very excited to immediately write back and accept. And probably a couple weeks later, I actually looked at the email and found out there was 22 minutes to do all that. So this is a challenge, but I want to provide sort of an overview of some of the cell and gene therapy approaches and immunomodulatory approaches that are available, put them in a framework, and try to make just a couple of major points about this that'll drive the discussion afterward and forward, and think about how all these approaches might work together toward a cure. Curing HIV is a tremendously difficult challenge simply because the virus integrates into these infected cells, and typically those are long-lived cells, as you've just heard. There's lots of them, 1 million, 10 million of them comprising the reservoir. And what this figure shows is that as soon as you stop some sort of suppressive therapy, the virus immediately comes roaring back, typically in 10 days to 2 or 3 weeks. I'm going to cheat and give you the take-home message right up here up front. There are a plethora of cell, gene, and immune modulatory therapies for HIV, and they're extremely promising. But they're not just pie-in-the-sky ideas. Many of these are already in clinical trials, and I'm going to highlight many of those. And some of them have already shown antiviral efficacy. So these are real interventions that haven't achieved cure yet, but have certainly had effects on the virus and on the reservoir. Here's the outline and sort of the framework we'll try to put these on. We'll first talk about approaches that allow us to reduce the reservoir, something that can actually take that 10 million cells comprising a reservoir and turn it into 1 million or 100,000 or 10,000 cells we have to deal with. And then the second part of the talk is, if we have this reduced reservoir that's there, a ticking time bomb ready to reactivate, are there ways that we can better control those reactivations that happen? And there's several approaches for that. And finally, we'll kind of wrap these things up with some concluding thoughts about how everything might tie together. The reservoir presents this incredible hurdle like I was telling you about, and Bob gave a very nice description of the viral outgrowth assay. This is a paper that Tae-Wook Chun did several years ago now. Essentially, if you look at a patient who was first treated during chronic infection, they typically have a fairly large reservoir. So if you do this is in the very right column, you can see, actually this is total DNA, but they have about almost 100 copies per million cells, so quite a bit of reservoir. If you take a patient who's been treated early after infection in the acute phase, they typically have smaller reservoirs, and this was a patient who got on therapy very early and then was suppressed for years and years, and had this vanishingly small reservoir. So a very logical question is, what happens should this patient go off of therapy? Will we have relapse, or has it decayed enough that they're cured? And the answer was, unfortunately, yes, we did have the relapse, or they observed the relapse. It was not within the 2 or 3 weeks that is typical, but maybe in 45 days, so maybe about 2 months, but relapse still occurred, and it looked pretty much like any other relapse. There was an initial control, but then the virus clearly escaped control and needed to be put on antiviral therapy. So this was pretty discouraging, and for a long time, many of us working on various approaches toward cure operated under this assumption we needed to get rid of every last virus if we wanted to achieve cure. And some of the hope, at least for me personally, came from a model that was developed by Bob's lab and published by Hill. Basically putting forward the idea mathematically, if you look at the size of the reservoir and ask, how does that relate to when the virus recurs? A typical reservoir shows recurrence very early in this 2 to 3 week time frame. But as the reservoir is smaller, at least predicted by the model, it takes longer for the virus to recur. And in fact, if you could reduce the reservoir enough by 2, 3, or 4 logs, you'd have extended times to relapse. Some patients would never relapse within their expected lifetime. So maybe we don't need to get rid of everything, we just need to get rid of most of it. So I think this was encouraging for a lot of people in the field. And it leads to this concept that, amazingly enough, hasn't really come forward in this session so far. So I thought for sure I would be totally redundant on this. But there's different ways we can think about cure. So we can think about a sterilizing cure, where there's no evidence of any virus anywhere in a patient's body by the most sensitive assays we can find. And they don't need antivirals, and they're truly cured. And then there's a second sort of cure approach or definition, which would be functional cure. And here, There's virus in the body. You can find it by sensitive assays. But in fact, it's controlled at very, very low levels. So the patient doesn't need to be on antiviral therapy. The virus is still controlled at a very low level. There's no threat of viral pathology, no threat of transmission. Essentially, this is functional cure, a very plausible and I think laudable goal. And then the third concept is remission, in which a patient might go for an extended period, in that condition, but ultimately relapse and then need to undergo more treatment or go back on antiretrovirals. So different concepts toward cure. So with that rubric then, how can we affect the reservoir? And actually we already know we can do this. So one of the things that happens in HIV infection is it predisposes people to leukemias and lymphomas, and one of the treatments for this is bone marrow transplantation or hematopoietic cell transplantation. So some HIV patients have required that because of their cancers. And we know that the myeloablative conditioning that occurs during transplant kills the cells that comprise the reservoir. But originally we didn't know how effective that was. And some of the early studies simply looked at patients who underwent transplantation. Here's a patient on ART with totally suppressed RNA on therapy, but a detectable reservoir by cellular DNA. They undergo transplantation right here, and their reservoir goes down to an undetectable level. Okay, so now is this patient cured of HIV? They go off of therapy, and in fact, the answer is no, they're not even close to being cured because they get a very rapid rebound of the viremia, and in fact, even a rebound of the DNA levels, or the RNA levels, back up to what looks like a primary infection. So transplantation alone is not enough to cure HIV. We know this has been shown over and over. Reduces the reservoir by perhaps on the order of a log. Okay, so tenfold on the diagrams from Bob's model, certainly not going to help us whatsoever toward achieving a cure. But when patients get transplants, they not only get myeloablative conditioning and then their own cells put back, but many get allogeneic transplantation. And one of the complications of that is graft versus host disease, where the transplant attacks the recipient cells. It can be a substantial problem. But another thing that's potentially good about it is that the reservoir itself is perceived as foreign by the transplanted cells. So there's a graft versus reservoir effect. And so one of the questions is, could allogeneic transplantation, if it's combined with suppressive antivirals, with this graft versus reservoir effect, could maybe all this put together lead toward a cure? And Bob mentioned the experience of the Boston patients, where exactly this happened. So these patients were transplanted. They achieved 100% donor chimerism here, and you can see that shortly after that, their DNA loads went to undetectable. So in these patients, there was no evidence of any provirus anywhere in the body that was detectable by these assays, even by an ultrasensitive assay much later in their course. They weren't able to detect that. So is the combination of conditioning plus graft versus reservoir enough to cure these patients? And I think many of you will know that the answer, unfortunately, was no. But it looks like things have been pushed out a little bit farther again. Again, thinking back to Bob's model, now you see that the delay from the interruption of therapy until the recurrence of virus is several months to over half a year. But then once the virus comes back, it comes roaring back. And again, control is lost. So graft versus reservoir can again substantially reduce the size of the reservoir, and probably more than conditioning alone, and probably now we're getting into the range of a couple of logs decrease. So how else can we directly affect this reservoir? As people are quick to point out, bone marrow transplantation is not going to be scalable. It's not going to be something we're going to offer to people. It has tremendous toxicities, and it's more proof of principle for this reduction of the reservoir. So one approach people have taken is to use immunotoxins, in which an antibody is conjugated to a toxin like ricin or other toxins, or radionuclide, and those can then be put into an infected individual, and in theory either attack HIV-infected cells that are expressing protein, or using other markers to attack those cells. Denton showed this in an animal model in which the bone marrow load of viral RNA was substantially reduced by about 3 logs, and similar reductions of lesser magnitude were seen in other tissues. Now this was all RNA focused. Okay, so this was able to identify and destroy cells that were actively replicating HIV. One of the questions that we don't really understand in the field is whether latently infected cells ever express enough viral antigen that they can occasionally be recognized, and over time one could get a decrease of the reservoir. So this might be exploited, though, if that were true, for the elimination of such cells. My own laboratory has been very interested in the concept of targeted mutagenesis of integrated provirus. We've been interested in a class of targeted endonucleases. Many of you will be familiar with these. We reviewed them several years ago now with Josh Schiffer. But zinc finger nucleases, tau effector nucleases, homing endonucleases or meganucleases, and then a lot of excitement recently about the CRISPR-Cas system. And one can direct these toward essential viral genes, and we've done this for herpes simplex, and we've done this for hepatitis B, and we're doing this for HIV. And if you do this for an integrated lentivirus, in this case with a reporter virus, you can get a dramatic reduction in the ability of the virus to express its genes, really down to the limited detection of the assay. So that's very exciting. In fact, this has been moved forward now with the development of the CRISPR system. That was all done with the meganucleases. With the CRISPRs, one can actually direct these toward the LTRs that flank the integrated virus and can very efficiently get excision of the virus. This was shown by Hugh et al. Last year in PNAS. And if you just look at two different types of LTR targeting CRISPR-Cas constructs, you can see they got a very substantial elimination of the virus and its ability to express genes. Now, a caveat of this, again, cure is going to require disruption of at least a substantial majority of the integrated virus. You know, if we can add another log to things, that would be great. But even one log, there's no in vivo delivery system right now that can even approach that in the relevant cell types. So I showed you this, I've alluded to this several times again from Bob's lab, this idea of the size of the reservoir determines reactivation. But in fact, we all know that once a virus starts to reactivate, various things can happen. And the analogy I kind of like to use sometimes is thinking about striking matches at a dry forest floor. That's a very different thing than if I'm doing it back home in Seattle in the rainforest. You know, we may or may not get a fire started, right? And so Alan Perelson's lab made a model that in many ways was similar to what Bob did, but they included a parameter that they called CTL killing rate. One can conceptually think of this as immune control in general, not necessarily mediated by CTL, or more generically, the ability of the immune system, or of the immune system actually to be infected and to support a reactivating infection, okay? So not only now would we want to reduce the size of the reservoir here on the y-axis, but can we somehow change things, making it harder for these rare reactivation events actually to take hold? And what this model suggested was that the combination of that gives you a much broader area of areas where you could have a functional cure, in which case maybe we would need less reduction of the reservoir if we made it more difficult for these rare reactivation events to take hold. So there have been a lot of approaches toward making T cells resistant to infection by HIV. In fact, this is much of the early work, and there's lots of papers here, and I can only hit a few, but people have used transdominant Rev constructs. They've combined those with antisense TAR, antisense ONV, ribozymes fusion inhibitors, and then combinations of these things. And I'll just show you a couple of these because they're sort of interesting. Here's a study by Morgan et al. Looking at RevM10 plus an antisense TAR. This is a human study. They didn't show an antiviral effect necessarily, but what they did show is that if they infused equal numbers of cells, either containing a control neo construct versus this transdominant Rev M10 and the antisense TAR, everything was equal very early on, but very quickly you saw a selection for the HIV-resistant cells, suggesting that they had an advantage in vivo. Similar studies have been done with antisense envelope. In this case, again done in humans, 5 patients. In this case, most patients had a fairly steady viral load after the treatment, but one patient did show over 1 log decrease in viral load after infusion of these cells, and that's depicted here as well. This construct of antisense envelope has gone into more extensive trials recently by Tabas et al. And again, showing evidence of effects. In this case, patients were treated with the antisense env and then put on a treatment interruption. And although most patients had relapses within the time that would normally be predicted, a few did have extended periods before they had reactivation. Again, this a hint that perhaps it has made reactivation more difficult to occur. Similar things have been done with an entry inhibitor. Now the idea is we'll make T cells resistant by making it hard for the virus to get in. There's an entry inhibitor called C46. This has been done in humans and resulted in these are patients, this is the average CD4 count of patients you see was decreasing before therapy. And then after therapy, there was a statistically significant trend toward a increase in CD4 count, so apparent therapeutic benefit. I should say in passing, and I don't have time to go into too much detail on this, but there seems to be a consistent trend in these studies that are protecting T cells by a variety of mechanisms that you get a much larger benefit than one might necessarily expect by protecting a few percent of cells. That those cells are probably augmenting the immune response, providing T cell help and other factors that allow the infection overall to be better controlled. And I think that's something that will need to be worked out over the next few years in more detail. Well, of course, we don't really need to do a bunch of gene therapy to get a situation where T cells are resistant. There's this natural mutation in CCR5, the CCR5 delta 32 mutation that prevents the virus from entering. CCR5 is dispensable for normal immune cell development and function. So in fact, a few percent of people, about 1% of Northern Europeans, already are homozygous for this CCR5 delta 32 mutation. It's less common in other populations, but this essentially prevents the ability of the virus to infect via the CCR5 pathway. And this is really the mechanism that led to the one really clear documented example of a cure of a person who had been chronically infected. And you know, this is a person who was actually here in the room just a minute ago. I don't know. There, hi, Timothy. How are you? I just saw you walk in. So Timothy was unfortunately developed an AIDS-associated leukemia and needed a bone marrow transplant. Unfortunately, was able to get a transplant from a donor who had this CCR5 delta 32 mutation. Now, a lot of things happened there. 2 rounds of conditioning, very dramatic graft versus host disease, and then a lack of susceptible cells if there was any virus that tried to reactivate. But the combination of all those led to a cure, and we don't really know the relative importance of all those. Unfortunately, this is a very rare mutation. It's hard to find these donors, and it really doesn't scale well. And perhaps most unfortunately, it's very clear multiple groups have tried to repeat this, and to my knowledge, it has not been successfully repeated yet. So our group and others at the Fred Hutch are looking at genetic ways for modifying CCR5. One could, in theory, do this by disrupting CCR5 within a patient's own cells. Again, you could go back to these targeted endonucleases that I spoke about. It can be disrupted either in mature T cells or in stem cells. And sort of the idea is you would take the person's cells out, treat them, and then put them back. If they were T cells, you'd reconstitute the T cell compartment. If they're stem cells, you'd get all this. And this has moved forward again into clinical trials. This is a paper that's published last year by Tabas et al., in which a number of patients got these gene-modified T cells and then underwent a treatment interruption. They all had recurrence, but there appeared to be a trend towards some of the patients starting to have a reduction in their viral loads toward the end of the treatment interruption. And by protocol, that had to end and the patients went back on therapy. One patient was actually negative at the time they went back on therapy. So you know, we were all very eager to see this play out, and there's a number of ongoing studies looking at this in more detail. Lots of excitement about a technology called chimeric antigen receptor T cells. Essentially, in this, an antibody binding domain is put together with sort of the back end of a transmembrane domain and then the CD3 zeta chain, so that when the antibody binds its ligand, the T cell is signaled to proliferate, to kill, whatever you want it to do. There's sort of different generations of this. They have increasing numbers of co-stimulatory molecules, and they progressively have worked better as they've gone from first generation to second generation to third. So the T cells now don't need MHC anymore. They can recognize any surface antigen on their target cells. This has been enormously exciting. I work at a cancer center and seeing this for anti-CD19 and other sorts of T cells for cancers, especially leukemias, just dramatic responses in otherwise treatment refractory leukemias. There have been clinical trials done for HIV. They're generally with the early generation CARs, sort of these guys back here. So there's not a lot of antiviral efficacy in those studies. But one thing that's very encouraging is that they last a long time. And this is a recent analysis of the 3 cohorts from Matsiasu, Deeks, and Aronson. And you can see if you look out a decade or so, you can see these things so they persist for an extendedly long time. So these will be great for controlling low-level reactivation. Going back to this concept, if there is occasional reactivation from the reservoir, there might even be an anti-reservoir effect. Vector delivery of antibodies has been really exciting. You can take AAV, put the coding sequence for a broadly neutralizing antibody or an antibody-like compound into those, shoot them into muscle. They'll stay there for a year or really years and continue to make antibodies. So Gardner et al. From Michael Farzan's lab had an antibody-like molecule that contained the CD4 binding domain, domains 1 and 2 with an IgG1 FC component, and then a peptide mimetic of CCR5. So this actually bound then HIV-OMV with very high affinity in a very broad manner, express this via muscle, via AAV, and macaques, and then sequentially challenge them with virus over time. And all the control cells or animals all became infected, but none of the treated animals did. So very, very exciting, thinking not only about now a challenge from the exogenous challenge, but actually just internal reactivation, a very good way to control that potentially. Immune modulation, we're sort of running out of time here. There's many, many approaches that are very exciting. Therapeutic vaccination toward conserved viral elements that the virus can't easily mutate. Subdominant epitopes. Jim Mullins is a very interesting vaccine in this regard. A lot of excitement about new adjuvants. You may have heard the varicella-zoster vaccine story. The new vaccine is 98% effective. It's amazing. Largely seems to be due to the adjuvant. Toll-like receptor stimulation makes a great adjuvant, TLR4, TLR7. There's reports of TLR7 being a latency reactivating agent. They're very interesting. Cytokines, I've only mentioned a few here. IL-15 is interesting for triggering NK function, increasing that. And then a lot of excitement now going back to the cancer field again, reversal of immune exhaustion. This PD-1 ligand axis, if that can be blocked, exhaustion can be reversed. Dramatic effects in certain types of cancer, and now this is being applied to HIV as well. So just to kind of put all this together in the last 30 seconds or so, I hope I've gotten across that there's sort of almost an unlimited number of avenues that you can put under this rubric of cell, gene, or immunotherapy. And they're not just pie in the sky, as I said. Many of them have gone into clinical trials, and a solid handful have already shown some antiviral efficacy. I do think that in regards to these, it's very clear no single one has achieved a cure, but I think it's probably naive to think about single agents for this. So I think the combinations are likely to be critical. And now, so leading into the quote debate, which is not going to be a debate, I think the combination with things like latency modulation, reversal of latency or suppressing latency, those are all going to really be important to play together. And then just to remind you that functional cure and remission are useful, and these may be laudable goals for us to go for in the intermediate term before we ultimately get to an ultimate cure. I just want to thank the various funders and supporters of the work who've helped us over the years, and thanks for your attention.